going off of Josh's presentation that explains the importance of having two antibiotics that had the ability to separate after being administered, I wanted to take a look at figure S10. So figure S10 is further emphasizing the importance of having the conjugates act separately, which have actually shown to produce some of the best results. So the scientists in this paper wanted to test the cytotoxicity of these conjugates as well as their different components when they were incubated within a macrophage cell. So in this test, the macrophage cells were tested with the different conjugates as well as their different components at a wide range of concentrations from 0 to 30 micromolars, which is shown here in this graph. And this graph is showing the percentage of cell viability, or the percentage of or the cell's ability to live and survive after being exposed to these different components and at different concentrations. So as you can see on this graph, there's actually a very high percentage of cell viability across all concentrations for almost all components. And what this tells us is that pretty much all of these components have very little cytotoxicity. And this is crucial information to this research because it tells us that these mammalian cells are not being significantly affected by the antibiotics like the bacterial cells were. So if we think back to Josh's presentation, where the P14-Con-S had the largest drop in bacterial growth, and we compare that to this graph where the P14-Con-S actually has one of the highest percentages of cell viability, and this is shown with the yellow bar across the graph. It's one of the highest percentages of the cell's survival, but I'm going to focus here on the highest concentration that was administered. And this bar right here showing the P14-Con-S is significant when we compare it to the bar next to it of the 1-to-1 mixture of the kanamycin and P14-LRR mixture. And this is, even though it's not that much greater, this actually is significant in this research, as it shows that the reducible linker is very important. Even at this the highest concentration that was administered, the P14-Con-S actually worked, was stronger and worked better at letting these mammalian cells live in these conditions. So having success with this P14-Con-S conjugate in vitro as well as in situ, or within a cell, the scientists wanted to see if the P14-Con-S would have the same antibacterial activity in vivo, or within a living organism. So what these scientists did was they chose C. elegans as their living organism model, and they first grew these C. elegans in the presence of P14-LRR, kanamycin, the P14-Con-S, as well as melittin. And they did this for 4 days. They grew these worms, and each day they monitored to see the percentage of the cell survival or the worm survival, in these conditions. And as you can see on this graph, at about day 3, there is 0% of worms that survived when they were treated with the melittin. And this is not very significant to the data, because we knew melatonin was shown to be toxic. So this was actually used as a control in this experiment. And as we see also on day 3, the P14LRR, the kanamycin, and the P14-Con-S did not have a very large drop in worm deaths, I guess. They all pretty much survived. And this was significant because this showed that the P14-Con-S was also not toxic in vivo within a living organism. So once they established that these drugs in question were not toxic within a living organism, they could now test the antibacterial activity within the worms without questioning whether or not the toxicity played a role in the conjugate's effect on the worms or on the bacterial growth within the worms. So, to test the activity of the drugs against a pathogenic bacteria, the scientists decided to infect the C. elegans with Salmonella enteritis. And then they proceeded to treat these infected worms with the P14LRR, the kanamycin, the 1-to-1 mixture of the two, and then the P14-Con-S. And they did this over a 24-hour period, making sure to check the percentage of bacterial growth within these worms. And what they found, or what they did, was they administered each drug at 3 different concentrations, at 10, 30, and 60 micromolars. And they did this just to see how different concentrations could or would affect the bacterial growth within these worms. And so, as you can see in this figure, the P14-Con-S had the most significant amount of bacterial growth reduction, with the highest concentration at 60 micromolars, clearing pretty much all but maybe 10% of the bacteria inside of the worms. So if we compare these results of the P14-Con-S with the 1-to-1 mixture of the P14LRR and the kanamycin, the P14-Con-S once again shows the significance of the reducible linker, as this conjugate works more efficiently on its own. And as we can see here on this graph, the yellow bars, even at its lowest concentration that it was administered, it's pretty much at the same, had the same effect as the 1-to-1 mixture did at its highest concentration. So this is just showing how strong the P14-Con-S was in clearing out that bacterial growth within these living organisms. So overall, the data for this figure shows that the P14-Con-S does have the ability to clear bacteria within a living organism. As we have learned throughout this presentation, there are many antibiotics today that are unable to effectively enter mammalian cells to target and clear out intracellular pathogens. However, the scientists in this paper suggest that this could be changing very soon. The use of a dual antibiotic with a reducible linker has showed to be very effective in targeting and clearing out these intracellular pathogens. And specifically in this paper, they focused on the P14-Con-S conjugate, and this conjugate showed very promising results within a living organism and could potentially be a very promising solution to these intracellular pathogens. So this P14-CoN-S pretty much suggested that this dual antibiotic approach can work, which is very exciting in this research. And so for the future, the P14-CoN-S conjugate's antibacterial activity should be tested and continue to be tested in different organisms. In this paper, only one organism was tested in vivo, which was the C. elegans, and within this organism, there was only one intracellular pathogen that was tested as well. So moving forward, there should be more tests with more organisms and different intracellular pathogens within those organisms to really test the limits of this system and of this process to see if this dual antibiotic approach is effective in a multitude of different situations. And I also believe that this, moving forward, this dual antibiotic process should be tested with a bunch of different antibiotics. It's been shown that bacteria have the ability to undergo mutations that allow them to resist antibiotics, allows them to not be affected by them. So a lot of different antibiotics should be tested, as this has shown that it could be a really big possibility that this works. But so overall, this dual antibiotic approach has shown that it does work, and shows a very promising future in combating intracellular pathogens in the future. Thank you for listening.